A warm good morning to everyone. Myself, Dr. Manasa D J, Assistant Professor, Department of Studies in Botany, Davangere University, Davangere. I'm very much glad to be a part of the 7th International Conference on Interdisciplinary Research for Sustainable Development. I would also like to thank the organizers for inviting me as a keynote speaker. I'm very much happy to share my research work on the use of nanoparticles in wastewater treatment. So, my presentation will be entitled as Musanda frondosa L phytochemicals mediated copper oxide nanoparticles and their phytocatalytic deterioration of the dye. Coming to the introduction part, as we know that in recent era, the synthesis of well-characterized metal oxide nanoparticles are of much importance to the researcher because of the extraordinary physical, chemical and optical properties inherent in them compared to the bulk material. Widespread applications of metal oxide nanoparticles in variety of fields including chemical sensing, electrochromic applications, catalysis have been observed. With this introduction, we also know that green chemistry has become a transpiring branch of chemistry which endeavors on biogenic, cost-effective, eco-friendly production of stable metal oxide nanoparticles which are profoundly studied due to its usual exclusive optical, electrochemical and catalytic properties. This methodology offers a rapid, cheap, eco-friendly one-step formation of biogenic copper oxide nanoparticles from the pool of reducing agents present in the leaf extract. As we know that in the modern era, textile dyes are significantly compromising the aesthetic quality of water bodies, which is going to increase the biochemical and chemical oxygen demand, which is going to impair photosynthesis, inhibit plant growth, enter the food chain, provide recalcitrance and the bioaccumulation, which may promote toxicity, mutagenicity, and carcinogenicity. So, with this background, we have designed the objectives of our present work. The objectives of the present work include to synthesize and characterize copper oxide nanoparticles from the leaf extracts of Musanda frondosa. The synthesized nanoparticles were subjected for dye degradation studies to check the efficiency of nanoparticles to degrade the carcinogenic dyes such as methyl orange and methylene blue. So this is the plant we have selected for our present work. This plant is Musanda frondosa, which belongs to the family Rubiaceae, which is a rambling shrub which is endemic to Western Ghats. This is the habit of the plant. This is the inflorescence, terminal inflorescence, and these are the fruits. The fruit is a berry, and these are the petaloid sepals bear by the plants. So coming to the occurrence of this plant is a rambling shrub which is endemic to Western Ghats, which can be found from Andaman, Konkan, Malabar and Tirunelveli hills. All along the Western Ghats, we may find this plant. Coming to the reason for the selection of this plant for the present study. So, we have made an extensive literature review on the phytochemical aspects as well as traditional uses of this plant. Traditionally, the leaves are used in the treatment of jaundice, asthma, hyperacidity, fever, ulcers, leprosy, wound and swellings. This plant has been investigated by several workers for its diuretic property, hepatoprotective property, then hypolipidemic, radical scavenging, antimicrobial, and it has also been used to treat fever, asthma and cough. So, flowers are being used in detoxifying mushroom poisons and terminate an early pregnancy. So, these are the pharmacological properties of this plant. Coming to the phytochemical aspect of this plant, Iridoids, flavonoids, and triterpenoids were found as an important bioactive compounds distributed in Musanda species. Phytochemicals such as astragalin, isoquercetin, etc. These are the flavonoids which are being isolated from the leaves of this plant. Several important other compounds like quercetin, rutin, heparin, ferulic acid, synaptic acid, saponins have also been identified by this plant. Keeping all this revealed in the mind, we have chosen this plant as a reducing agent for the synthesis of nanoparticles. So coming to the methodology, first step will be the collection of plants from the Western Ghats. Later on, the plant extract will be prepared. Then the extract will be subjected for polyphenol quantification and antioxidant activity determination of the extract. Later on, based on the phenolic compounds as well as antioxidant activity exhibited by the Musanda frondosa leaf extract, we have chosen the leaf extract for the synthesis of the copper oxide nanoparticles. The synthesis was carried out by solution combustion synthesis method. We have followed the method which was followed by Meghana et al. 2020. Upon the synthesis of nanoparticles, characterization was done. Later, the nanoparticles were subjected for photocatalytic degradation studies. Coming to the results of the present work. So, once the extract was prepared, it was subjected for quantitative analysis of phenolic compounds present in the plant extract. As we know that polyphenols are important constituents of plant derivatives, the aqueous extract of Musanda frondosa was found to possess total phenolic contents in the range of 43 to 79 milligram per gram of the plant extract. So the reducing power of the Musanda frondosa was evaluated and compared with the standard ascorbic acid. At the lower concentration, the Musanda frondosa extract had enhanced the reducing power capacity owing to its ability to reduce the metal precursor to corresponding metal oxide nanoparticles and stabilize them. Here the graph is corresponding to the reducing power assay, which corresponds, and the picture here shown represents the corresponding color change with an increase in the concentration of plant extract. With the increase in the concentration of plant extract, the activity exhibited by the plant is also increasing. So the comparative, coming to the FTIR analysis, once the we have found that polyphenol components was present and the antioxidant capacity of the plant extract was determined and the IC50 value was found to be 183 microgram per ml. So with this result, we have selected the plant extract for the synthesis of nanoparticles. Once the nanoparticles were synthesized, characterization was done. First characterization technique was FTIR analysis. Here we are having two graphs. This graph represents the IR spectra of Musanda frondosa crude extract, and this is the expanded form of IR spectra of copper oxide nanoparticles. This gives you the comparative analysis of the FTIR spectra of the plant extract as well as the nanoparticles synthesized from that extract. The leaf extract showed several prominent peaks which might be attributed to the functional groups of the phytochemicals present in the leaf extract. These phytochemicals play a vital role in the reduction of metal precursor salt to corresponding metal oxide nanoparticles. This peak, which is present around 3004 168 centimeter inverse is due to the alcoholic or phenolic OH stretching modes which is present in phenolic as well as flavonoid groups. Likewise, we have found various functional groups which might be present in the as a flavone derivatives, proteins, and other phytochemicals. So, similarly, here we have observed a small shift in the observed peaks in the copper oxide nanoparticles IR spectra. These um, peaks corresponds to the phytochemicals which were responsible for the capping of the nanoparticles synthesized, and the strong peak which is present at the wave number around around 500, 545 and 530 centimeter inverse, which is because of the formation of copper oxide nanoparticle. Then coming to the powder X-ray diffraction studies, which determines the structural properties, crystalline phase determination will be done using this powder XRD analysis. Here we have observed various peaks which are well indexed with the corresponding 2 theta and favored orientations of the planes at 32, 35, 38 and 48, 53 and so on. This is with reference to the JCPDS card number 80 1916. The spectra is in good agreement with the monoclinical structure of the copper oxide nanoparticles. So no additional diffraction peaks were observed, which indicates the high single phase crystal purity of the prepared nanoparticles. It is evident from the XRD patterns that the peaks are sharper and narrower at the base. In this second image, we will be able to observe the peaks are sharper. The peaks are sharper and narrower at the base and becomes broader as the reducing agent volume was increased. There is a shift in the peak position. You may observe here there is a slight shift in the peak position with an increase in the volume of the plant extract to obtain the copper oxide nanoparticles. Coming to the SEM analysis. So the surface morphology and their elemental composition of the biogenic copper oxide nanoparticles were analyzed using SEM and EDS micrographs. The distribution of the nano-rigid particles is not homogeneous. It appears to be agglomerated. In all the images, you may observe the nanoparticles are found to be agglomerated because of the use of plant extract as a reducing agent. Then the surface morphology of the synthesized nanoparticles are found to be spherical in shape at higher magnifications. These are the EDS spectra of the nanoparticle synthesized, which confirms the formation of copper oxide nanoparticles evident from the copper as well as oxygen signal peaks along with the carbon peaks. If we can observe the carbon peaks also in the following image, which is due to the base used for the analysis. Okay, that is because of the presence of phytochemicals. Coming to the UV visible spectral studies of the copper oxide nanoparticles. The optical properties of the sponge-like porous nanostructures were determined by UV visible absorbance spectroscopy. UV visible spectra display a broad peak ranging from 370, here you can see 370 to 390 nanometer, which is due to the surface plasmon absorption of metal oxide. The metal oxides exhibit surface plasmon absorption, which can be attributed to the collective oscillation of the, which is due to the collective oscillation of the free conduction band electrons, which gets excited due to the incident electromagnetic radiation. This results when the incident light wavelength predominates the particle diameter. The absorbed surface plasmon absorption with a maximum at 378, 391 and 370 nanometer for different concentration of the plant extract used. The optical band gap of the synthesized nanoparticles was evaluated by the fitment of the absorbance spectral data to the torque relationship, and the band gap was found to be around 3.96, 3.6 and 3.47 electron volt, corresponding to 5 ml, 15 ml and 25 ml of the plant extract used for the synthesis of particles. Coming to the dye degradation studies, upon the confirmation of nanoregime of the synthesized particles, we were curious to know about the efficiency of the synthesized nanoparticles for the degradation of the dye. So we have selected two different dyes. One is methylene blue and the other one is methyl orange dyes. In the first figure A, you will be able to notice the photocatalytic efficiency was found to be around 97, 92 and 92% for 5, 15 and 25 ml of extract. Similarly, from the figure D and E, we may observe the efficiency rate was around 94 and 88% for 5, 15 and 25 ml of extract for methyl orange. This is for methyl blue and this is for the methyl orange dye. The results here you will be able to identify the kinetic study of the reaction was also studied, and it was found that it fits under first order kinetic reaction. So these results favor the nanoparticles to have very good efficiencies due to the smaller size and optimum band gap values. This slide shows the effect of catalyst dosage and initial dye concentration. We have conducted a series of experiments to evaluate the optimum catalyst dosage for the effective degradation of the dye solution. The percentage degradation rate was found in each case, and a comparison graph was plotted to know the optimum dosage of catalyst for effective degradation of methyl blue as well as methyl orange dye. From the figures, it is evident that the catalyst reaches a optimum level of around 20 to 30 milligram, after which the degradation efficiency decreases. It was noted as the degradation rate decreased significantly with the increase in the amount of the catalyst loading beyond 30 milligram, which may be because after certain contact time, the absorption sites were filled and the catalyst was found to be floating above the solution during the time of investigation. So the effect of initial dye concentration on the dye degradation rate percentage was studied from the range from 10 to 50 ppm aqueous solution of both methyl orange and methyl blue. So the huge amount of dye gets absorbed on the active sites, thus blocking the contact between the radicals generated from the conduction and valence band of copper oxide nanoparticles on the irradiation of the light source. So we were curious to know about the recyclability of the nanoparticles once they are used for the treatment of dyes. So we have studied the recyclability profile of nanoparticles. For the practical use of these nanoparticles, it is very essential to perform the recyclability test. Hence, an effort was made to understand the reusability of nanoparticles by using it repeatedly over 5 cycles. You can observe here in the graph number of cycles. We have used the same catalyst for 5 consecutive 5 cycles for the degradation of dyes. This is for methylene blue and this is for methylene orange. Okay, so about 10 milligram of catalyst was used to study the recyclability of copper oxide nanoparticles for the degradation process. Recovery of nanoparticles was carried by adopting simple centrifugation method at high RPM. Okay, it was found that all the nanoparticles effectively or efficiently degraded the model dyes used successfully for 5 cycles. Even after the use of 5 cycles, they have still retained the capacity of degrading the uh, dyes. So all the nanoparticles were found effective more than about 72 and 82% in the degradation of methylene blue and methyl orange even after fifth cycle of their use. There is a slight decrease in the photo. Here you can see the percentage degradation of dye is slightly decreasing. So there is a slight decrease in the photocatalytic efficiency rate after each successive cycles. This may be due to the loss of photocatalyst by weight after the recovery process. So coming to the conclusion, we have synthesized the nanoparticles using the Mosander frondosa phytochemicals by solution combustion synthesis method. It was found that on the varying volume of plant extract, there is an influence in the size of the nanoparticles on keeping the temperature and other parameters constant. And we have done some characterization, we have used some characterization tools for the confirmation of the nanoparticles which were obtained. All the synthesized nanoparticles were found to have good photocatalytic activity. They were effectively degrading the carcinogenic dyes with 90 to 95% of photocatalytic percentage dye degradation, and the recyclability profile of the dyes was also very effective. We can use the same catalyst for successive 5 to 6 cycles for the degradation of dyes. Coming to the future scope of this present study, because of their properties of oxidation, precipitation, reduction and absorption, nanoparticles can be applied worldwide for the removal of contaminants, sewage treatment such as nitro-aromatic compounds, inorganic anions, phosphates, radio elements, organic dyes, etc. So this present study was based on micro scale experiment, but this could be done for industrial scale wastewater treatment also. An incubation center can be set up for the degradation of this catalytic dyes. And I would like to acknowledge DSD for awarding me Inspired Fellowship with which I could able to finish this project. And also I would like to acknowledge the great and immense support and encouragement of my research supervisor, Professor K.R. Chandrasekhar. And these are our publications, these are the recent publications wherein we have reported the synthesis of nanoparticles from the callus also, as well as the crude plant extracts, and we have studied the photocatalytic dye degradation as well as the anti-cancer properties of the synthesized nanomaterials. Once again, I would like to thank the organizers for giving me this opportunity. Thank you so much.